we are on. It always takes a sec. Yeah. All right, welcome back to Ask a Scientist, everyone. My name is Abby with the Asheville Museum of Science, and today we are going to be talking about how scientists can use organic chemistry to treat diseases. And so today I have one of my favorite professors with me. This is Dr. Chris Monceaux. He is a professor of organic chemistry, among other things, at Radford University, my alma mater. So I'm really excited to have him here with us today. He got his bachelor's actually at Radford. So he's a Radford alum, and then he continued his education at NC State and then his PhD work at Virginia Tech before making his way back to Radford because he just couldn't stay away. So I'm going to go ahead and pass it off to Chris Monceaux, and he can take it away to tell us all about his work with organic chemistry.
All right, thank you for that introduction, and I'm glad to be here. Thanks for the invitation as well. I would like to go ahead and share my screen, if I can find my mouse. Where did it go? I think I'm stuck. I can't find my mouse. This is embarrassing. Okay, so are you seeing my screen now?
Nope.
Okay.
See your face.
All right, I think I got it back. All right. Desktop. You see my desktop now?
Mm-mm.
Okay.
Oh, there you go.
Okay, great. Sorry about that.
That's okay. Classic Zoom.
Yes. All right.
Everyone go ahead and send in questions along the way as well, and we'll get Dr. Monceaux to answer them.
Okay, as Abby said, we're going to be talking about using organic chemistry to tackle diseases. And before we get into the discussion, as Abby already said, we're both alumni, I guess you can say, of Radford University. So where is Radford? It's just north of Asheville and across the border in Virginia, Southwest Virginia. So we're in Appalachia. We are in the city of Radford, which is on the New River Valley. And I'm presenting from the chemistry department today. And we have about 110 students. And we are spread across 3 different buildings on campus, and we have a lot of modern instrumentation and spectacular lab space to do some of our research in. So give us a look if you're ever interested in contacting any scientists here, or if you're interested in never attending Radford University. So what I want to get into next is looking at what we can do with organic chemistry. Now, not everybody watching this has probably taken organic chemistry. It's been a long time. Or you are an organic chemist, but nonetheless, I'm not going to get too nerdy with organic chemistry today. But a lot of times, organic chemistry is kind of treated as like the bad science, right? It's the one that almost kept me out of medical school, or every time I go to the dentist, they're like, "What do you do?" I'm an organic chemist. Like, oh, so, yeah, there's a lot of a bad pub out there for organic chemistry, but I'm obviously biased. I love it. And what really got me interested in organic chemistry was actually started with plants. So I began after getting my master's degree at the Department of Agriculture. And what we were doing there is we were looking for anti-mosquito compounds that were coming from plants. And what really got me interested about organic chemistry there is that at the time, it was just a course I took in college. I didn't really like it all that much. But then when we started looking at, all right, extracting molecules out of plants, and then putting in these biological assays and seeing how they were affecting mosquitoes, I was like, wow, so you can actually look at molecular compounds and see how they interact with life forms. So I eventually moved from mosquitoes into humans and got interested in what I'd call, kind of CNS or neurological chemistry. So looking at how compounds can affect neurological processes in the body. So one place I started when I got to Radford University was looking at epilepsy. So on this slide here, we can kind of define epilepsy as convulsive or non-convulsive seizures. All right, and this is a neurological disorder that affects persons of any age. So things like Alzheimer's disease that we'll talk about a little bit later, that tends to target the elderly population, whereas epilepsy really doesn't discriminate based on age. And about 50 million people across the world are affected by epilepsy. So what can we do to help this? Well, as an organic chemist, one thing we look at are drugs. All right, so we make synthetic compounds to help alleviate some of these diseases. And one place we can do that is in these anti-epileptic drugs. All right, so there's really three mechanisms that these AEDs or anti-epileptic drugs work. All right, the one that's on the market now for this is Lyrica, and we're not going to talk about how that works, but it is essentially a voltage-dependent ion channel modulator. So it affects how ions move across the cell, the nerve cell, and how that is then working for neurotransmission. The one I want to talk about is the synaptic inhibition enhancers. All right, so this is how we're going to get the neurotransmission to work in our favor. So in the very bottom here, I have the molecule gamma aminobutyric acid. So that is GABA, right? That is a neurotransmitter that is naturally produced in your body, all right? And that is what sends the biochemical signal, essentially. It's involved in sending a biochemical signal. So we're looking at that molecule there, and it's just called GABA. It's very simple, all right? And it is. And how does it work? Well, it works in a much more complicated system. And what I've got here from our organic chemistry textbook is a picture of a nerve cell. All right, so we've got that whole cell in blue, all right, and it has one end over here on the left, and then it runs all the way to here on the right to another nerve cell. Now, these two nerve cells are not in actual contact with each other, all right? So what we have is an area of detail below, and this is what we call the presynaptic neuron on the left, and then we have a postsynaptic neuron on the right. So what ends up happening here is we want to send a biochemical signal through our nervous system. We have to do it through these nerve cells. So you want to kind of think of them as wires, all right? So we're transmitting electricity from one end of a wire to another. But in the case of wires, they're physically contacting each other and the electrons flow through them. In the case of nerve cells, they're not physically contacting each other. They are actually separated by something called a synapse, all right? And what we have to do is send a neurotransmitter, which we're showing in this red dot, and it needs to be released from the presynaptic neuron over to the postsynaptic neuron. So the way that is released is basically through osmotic pressure, all right? So what happens is that presynaptic vesicle gets squeezed and out pop the neurotransmitters. And then they go across the synapse and they arrive at the postsynaptic neuron and they arrive at something called a receptor. And then that receptor recognizes the neurotransmitter and then fires a signal on down the neuron to the next neuron. So we just kind of see this cascade that occurs. So one question is, all right, what happens when we have something like a seizure, right? So in the case of that, that's where we're looking at this GABA system, all right? So the nervous system is made up of different types of nerves. You have your GABAergic nerves, your serotoninogenic, so right, so those with the serotonin, and then you have your dopamine, let's see, acetylcholine. So there's several different types of nerve cells. The one we're focusing on right here with the epilepsy is the GABA. So it's found that people that suffer from these seizures are lacking in something called GABA, right? So GABA is that neurotransmitter. So we don't have enough of these red dots, essentially, for people that are suffering from these seizures. So how can we get more red dots, all right? So the natural approach to be like, well, can't we just give them more GABA, all right? And as you saw, GABA was the simple neurotransmitter that's naturally made. So why can't we just, sprinkle that on your cereal or something like that? But the problem is it's such what we call a hydrophilic molecule, all right, that it will just be kind of washed out like taking too much vitamin C. It'll never get to its target, all right? So we have to get it into, tissues. It has to get across this blood-brain barrier. There's all these complicated processes that the compound has to get to its target. So you have to kind of balance the what we call drug design. You don't want it to be too hydrophobic, right? Then it'll just get stuck and it won't, in tissue, it may never cross the blood-brain barrier. And then, of course, you don't want it too hydrophilic where it just gets flushed out. So we got a question. What's up?
Yeah, I was just going to ask if you could briefly explain how hydrophobic versus hydrophilic compounds act in the body.
Okay, sure. So one of the easiest places to go to that is in your vitamins. All right. So you can look at a multivitamin that you may take. One vitamin that you typically will see in a multivitamin is the vitamin D. All right. So vitamin D, if you look at its molecular structure, it has a lot of what we call polar groups. So it kind of, has things that water likes, right? So water we know is just H2O. We have OH bonds in water. All right. So vitamin D has a lot of these polar groups. So what that means is you don't really have to worry about taking too much vitamin D because it essentially gets flushed out in your urine. All right. So sometimes you'll see these, vitamin B complexes you can take, and it's like 1,000% of your recommended daily value of vitamin D, and that's fine. But if you look at another vitamin like vitamin D, all right, that is much more what we call lipophilic or hydrophobic. So it doesn't like water as much; it likes fatty tissue much better. And you can get too much of that, right? And the problem there is since it's polar, it gets stuck in your tissues, doesn't get flushed out as well. In fact, what is the compound? Beta carotene, right, is another one, right? That's what makes carrots orange. So you can actually drink carrot juice, and if you drink too much carrot juice, your skin will actually start turning orange, right? That's because all that beta carotene is getting stuck in your fatty tissue, and you can actually see it through your skin. So did that help out a little bit? All right, good. So again, what is our mission here? Our mission is to get more red dots in between the synapse. And one way we can do that is by knocking out the activity of enzymes that are responsible for kind of policing the synapse, all right? So we have these naturally occurring proteins, and I like to think of them as kind of like little Pac-Man, all right? So if we look here, it looks like Pac-Man, and then it has something called a substrate. So the substrate in this case would be GABA, all right? So what this enzyme is responsible for doing is swimming around, looking for GABA, chomping down on it, and basically cutting it in half. All right. So it causes some kind of chemical reaction that breaks GABA. All right. So, whether it be a hydrolysis reaction, something like that, it just ripped it apart essentially. And then those GABA fragments can be reformed somewhere else, but essentially they're taken out of the synapse. So why do we have these? Well, if you have too many of those red dots, then you've actually got the opposite problem, right? So you can have too little of the red dots or too many of the red dots. You want just the right amount. So that's what this enzyme is supposed to be doing, regulating that just right amount. However, people that suffer from epilepsy don't have the right amount of neurotransmitters. So you would imagine that an enzyme is making the problem worse. So what we can do is we can actually give the enzyme, kind of trick it. We give it what we call an inhibitor. This is a synthetic compound that will compete with the substrate, all right? So we have this competitive inhibition. So Pac-Man is to choose between the substrate and the inhibitor, all right? And what ends up happening is that the inhibitor has a very strong binding profile to the enzyme, and it lets go of it very slowly, all right? So what ends up happening is Pac-Man gets tied up dealing with this inhibitor before it can spit it out, all right? Now, the idea is that this is a reversible process. Pac-Man will eventually give away that inhibitor, and then it can go back to chomping up GABA if it wants. But if you tie up Pac-Man, keep him busy, you can have more GABA in the synapse. So these type of drugs are what we call enzyme inhibitors, all right? And that's precisely how the drug Sabril works, all right? That's its trademark name. So up here, what I'm showing is the natural substrate GABA. And then we have this GABA-T, which is the enzyme that's responsible. So this is the Pac-Man that's going after GABA, the natural substrate. But there is an inhibitor that Vigabatrin, or the trade name Sabril, it looks a little bit like GABA, right? So that's why the enzyme will recognize it, like, ooh, a little ghost to eat, right? But this part of the molecule over here, for reasons that I don't want to get into, will actually cause slow release of that substrate, right? Or slow reaction of breaking down that substrate. Yeah, question.
Yeah, we had a really good question from Ben Gelheiser come in asking, is that enzyme specifically targeting the GABA, or is it policing other things as well? Would it keep it tied up potentially, would keeping it tied up potentially cause other side effects?
Yes, and there are that enzyme has a very specific role, and that's a good thing that nature has developed. So enzymes are very specific for what they're supposed to do, and I'll show you some images of an enzyme later. So there's kind of this lock and key theory, right? That the enzyme is looking specifically for that one substrate. And one problem with this is that if we have an irreversible inhibition, which I'm not showing here, what that means is that the enzyme will never let go of the inhibitor, right? You make a covalent bond essentially between the enzyme and the inhibitor, and you never let it go, and that enzyme is wiped out forever, right? And the problem is you don't make enzymes that fast. So if you start wiping out too many enzymes, you're going to start having irreversible damage, and that's precisely how sarin gas works, or VX gas, right? Those are irreversible enzyme inhibitors for an enzyme called acetylcholinesterase. All right, so essentially, you want to have a compound, though, the inhibitor. You want it to be selective for just a few enzymes. All right, unfortunately, these inhibitors often are not selective, and there's other enzymes that we have to compromise in the process, and that's all looked at during kind of toxicology studies and so forth. Are we knocking out this enzyme? Are we knocking out this enzyme? Well, that's extremely important. So you've got to make sure that your inhibitor is really selective for the enzyme that you're targeting. Does that answer the question? All right, good. Good question, thanks. All right, so what I want to move on to now is not just looking at enzymes, but looking at something called reuptake. And this is a well-known therapy. And I'll get into where else we've seen this before. So again, I've got my cartoon here where I have my two nerve cells, all right? And I've kind of blown up this area as our synapse, and I've got on the top my presynaptic neuron and my postsynaptic neuron on the bottom. And then we see, instead of little red dots this time, the black dots. Those are my neurotransmitter, and in fact, this is my GABA. And what they're doing, they're getting squished out, and they go into the synapse, and they've got to get to this receptor on the postsynaptic neuron. So my receptor is this little blue pentagon, all right? So we, of course, talked about the enzymes kind of policing this to make sure there's, enough GABA, the right amount. And of course, if you're suffering from GABA, you may want to knock out that enzyme or kind of slow it down. But something else that happens in the body is we have these reuptake proteins that are sitting on the neurons. And what I want you to think of those as is kind of like, pumps that pull excess neurotransmitter back into the presynaptic neuron. So it's kind of like if there's too much in here, they're like vacuum cleaners. They kind of clean up that, and they recycle it. So again, if somebody's suffering from epilepsy, they may not have enough of this neurotransmitter in the synapse, and having these reuptake proteins may be harming that, right? Because if you're sucking them back in and they never get to the receptor, that could be a problem. So what we can do is we can actually slow down these reuptake proteins just like we were slowing down the enzymes, all right? So in fact, in the GABAergic system, we see 4 different types of these reuptake proteins, and they're called isoforms, all right? And we're not going to get too deep into that, but the major one that's targeted in anti-epileptic medicine is this GAT1 isoform, right? So that's the one that's expressed most often. In fact, they have a FDA-approved treatment to inhibit this reuptake protein. So reuptake inhibition is not only used for GABA, but it's also used in depression and anxiety medicine. So there are these drugs called SSRIs, all right? And these are the selective serotonin reuptake inhibitor, all right? So what they do is they allow more serotonin throughout the nervous system by inhibiting the reuptake of serotonin, all right? So, Zoloft and Prozac and all those kind of compounds or SSRIs. All right, so where did we start? We've talked about, the biochemistry, what we want to do. We want to block these reuptake proteins. So where do you start? Well, you most often start with the natural substrate. You take your tip from Mother Nature and you say, look, you've designed this ligand to, go into the reuptake protein. Can we find synthetic modifications that will also be recognized by the reuptake protein, but let go of the reuptake protein slower? All right. So again, it's kind of keeping that reuptake protein busy while GABA gets to swim around and do its job. So at the very outset, there were some studies done on these 3 molecules up top, right? And they're very small molecules and they look a little bit like GABA. So what I want to kind of point out that GABA was this gamma aminobutyric acid. So on one side of the molecule, we see this NH2, and on the other side of the molecule, we see this, what we call acid group. All right. So what we can do is we can start looking at, all right, can we modify that, but keep some of those properties intact? And that's what these 3 molecules did. And they actually did a great job at inhibiting the reuptake of GABA. However, these again are very small, what we call hydrophilic molecules. So these are going to be like your vitamin Bs, they're just going to get washed out and they're not going to be very effective in vivo. They were very effective in a laboratory study. So what some drug companies did is they took those 3 and they built 2 molecules, one of which here on the left is called tiagabine. That one's actually FDA approved. You can get a prescription for it if necessary. But where does this come from? Well, we see this part again, all right, that ring system with the acid group, and then we have our nitrogen over here. But then we have this thing, kind of dragging off to the left. And that's what we're going to call this hydrophobic tail, all right? So that makes it kind of water-fearing, all right? And it gives it that kind of Goldilocks balance of being polar and non-polar, so it can kind of travel to where it needs. And another compound that was successful, this was a research compound, it's actually not FDA approved at this time, it's this research compound EF1502, right? So both of these were very effective in the reuptake of GABA, and tiagabine more so is actually shown to be effective in the body. So I kind of want to talk about what these reuptake proteins look like. All right. So this is a cartoon, and we can kind of call this the topology, if you want, of the GAT1 transporter. All right. So what we're looking at here is a cross section of a nerve cell. All right. And this would be on the outside of the nerve cell. So this is towards the synapse, and this is inside of that nerve cell. So that's towards the inside of the cell. So we have these proteins, this protein that's kind of crisscrossing, going through in and out of this cell membrane. And what I'm showing here in yellow is this area of detail, all right? And there's 2 little things I want you to pay attention to inside this area of detail. There's a triangle with an L, that stands for ligand, all right? So ligand is something that binds to the protein, all right? So this guy is our what is binding to the transporter. And then we have this little blue dot, and that blue dot is a sodium ion, all right? So inside of this active site of the transporter, all right, so this is where recognition happens. If you kind of come in with the sodium ion and the ligand is recognized by these protein domains, you're allowed in, all right? If you do not fit that mold, you're never going to get in, all right? So the idea is that GABA would come in as the little L with the sodium ion, has the correct password, and can get into the cell, all right? If we wanted to stop this process from happening so quickly, what we could do is we could fake out this gatekeeper by having a synthetic modified version of L, and it would come in and it would really confuse this domain for a little while, and then it would let it go, all right? So that's the idea. And, of course, the protein's not alive. We're not really confusing it. We're just taking advantage of certain types of bonds that we're going to talk about here in a moment, all right? So how in the world do we confuse this protein? Well, what we can do is look at this binding site, this area of detail. And again, I've talked about this sodium ion. So why is the sodium ion necessary? Well, the sodium ion is necessary to kind of make the shape of the domain correct. All right. So all the proteins pull in to that sodium ion. How do they pull in? All right. So you may have learned something back in chemistry, intermolecular forces. All right. So we have this red thing here, that's oxygen, and this blue thing here, sodium. All right. So we have oxygen, which is electronegative. It's attracted to this sodium. Right. So you've got this nice barrel being formed from the intermolecular reactions of the protein and that sodium ion. And once you have that shape formed, then our ligand, which is in yellow here, can bind to the sodium, and now it's allowed through. All right. So what we have to do as organic chemists is we have to look at, well, what can we design to interact with this protein? All right. So the protein is shown here with these green ribbons and this red ribbon. Those were those domains I was showing you outside. And then these, or on the previous slide. And then we have these letters and numbers. What the heck do they mean? Like A22. All right. That is a specific amino acid residue on the protein. All right. So this would be alanine 22. All right. So then we see tyrosine 254. All right. So we have these different amino acids that are, binding to that sodium. And what we can do is we can design a drug that would fit in here, but let go much slower. So that's the whole idea, all right, behind the inhibition of a transporter protein. So what are we doing here at Radford University? Well, we're again taking a tip from previous research. All right, so we're looking at this compound that showed efficacy in laboratory tests, not in vivo, this ACHC, and we're doing synthetic modifications of it. So this is currently ongoing in our laboratory. All right, we have students working on this. I keep about 3 to 4 students in my lab working as undergraduate researchers. So they have hands-on experience as undergraduates, doing multi-step synthesis. So they're doing their organic chemistry. They're trying to make these compounds, and it's a work in progress right now. All right. So the next one I want to get into is Alzheimer's disease. All right. So Alzheimer's disease is, pretty much classified as, a neurodegenerative disease. All right. So there are 2 real targets in these, in neurodegeneration. It's these either what we call tau proteins or these neurofibrillary tangles. All right. Or we can have these amyloid plaques. All right. And these plaques can get in between the neurons in the brain, and it really clogs things up. And of course, then we see neurodegeneration. So while I was at Virginia Tech, we did some research on targeting the what we call the generation of these amyloid plaques. All right. So where do these amyloid plaques come from? So this is another cartoon, and what we're looking at here is the cross section of the nerve cell yet again. All right. So the cytosol, that is the inside of the cell, and then we have the lumen, which is the outside of the cell. All right. So we have this protein right here in the middle. It's called APP. All right. It's what's called the amyloid precursor protein. And there are 2 enzymes that act on it. All right. There's what's called the alpha secretase, and there's another enzyme called beta-secretase. And those 2 enzymes actually clip the protein at different places. All right. So the protein that's sticking up out into the lumen is cut at different places. So alpha, we call that the alpha secretase pathway, and this doesn't cause any plaque development. However, beta secretase, unfortunately, does. All right. So if we look at this cascade, once beta secretase does its job, then another enzyme called gamma secretase makes another clipping of the protein, and then we release this one protein called A-beta 42. All right. And A-beta 42 is shown to kind of make these aggregates and then form plaques. So the idea is we don't want A-beta 42 developing at all. So how can we stop them? Well, we can either stop gamma secretase from doing its job, unfortunately, gamma-secretase is necessary for this pathway. So, and it's actually shown in mice studies that if you knock out gamma-secretase, you're in trouble. All right. But another option is looking at beta-secretase. All right. So the inhibition of beta-secretase. So this, again, we're talking about enzymes. So this is another Pac-Man that we're targeting here. We're targeting that beta. This is what it kind of looks like. All right. So earlier we were just talking about it's a Pac-Man. This is an actual computer-generated drawing of that compound. And what we're looking at here is a kind of a space filling of this protein. So in green, you're seeing carbon atoms. In blue, you're seeing nitrogen atoms. Red is oxygen, and yellow is sulfur. All right, so these are the most common elements that you're going to see in protein structure. All right, hydrogens are not shown because you wouldn't see anything but hydrogen if that was the case. All right, so this is the beta-secretase enzyme. So this is the bad guy. This is the one we want to go after. All right, but if you look closely in the very center, you'll see something that kind of looks a little bit different. All right, that's the ligand that's actually in there. And if you put a ligand into this beta-secretase, again, you're giving it an inhibitor and you're slowing down the operation of that beta-secretase, or what we call the base 1 enzyme. All right, and if you slow it down, you're not going to make as many plaques. So how do we make a ligand? All right, so there's this idea in medicinal chemistry, drug chemistry, that we call structure-activity relationships. So in red, I have this molecular structure, all right, that was designed by a chemist at Merck back in 2006. And what they were able to do is make this cartoon where they show key amino acid residues in the enzyme making intramolecular bonds, all right? So if we look here, this is just a simple hydrogen bond, right? And that's not a covalent bond, all right? So that can come on and off. Then we have over here a hydrogen bond, another hydrogen bond, another hydrogen bond, right? So these aren't actually what we call covalent hydrogen bond. This is that hydrogen bonding that you've probably heard of in many of your science classes, all right? And there's an old joke, if you don't know the answer to a question in organic chemistry, you just say hydrogen bonding, all right? And it's such a big deal. I actually have a book, a whole book on hydrogen bonding, all right? So it's that important. But anyway, back to this, we see all the different hydrogen bonding, and that is what causes that molecule, the inhibitor, to fit into the enzyme. It's just like, ooh, look at all these nice hydrogen bonding interactions, and there's so many of them, I really can't let go, all right? But eventually it does let go, all right? But you slow it down. So it binds much better to the enzyme than the natural substrate would, all right? And again, at Virginia Tech, we were doing some of this research, so just showing you some of the compounds that we synthesized based off of that structure, all right? And they showed pretty good activity against that base 1 enzyme, all right? So maybe a question many of you are having is, can we use organic chemistry to attack this coronavirus that's going on now? And just probably in the last couple of weeks, I came across a paper where they were looking at a drug target for the coronavirus. So this is part of the coronavirus's replication system here. So we're seeing this kind of crown-like protein that sticks out on the outside of the virus. And what it does is it's responsible for basically shooting out RNA, and then that RNA is then replicated within the cell, and then we get more virons built up, and then we have the infection. So if there's some way you could put a drug down into that opening, then you're going to not allow the RNA to shoot out. So, is this going to happen anytime soon? Probably not, but it's at any rate, even, drugs, that we've talked about in the central nervous system can be used way other places with the same kind of organic chemistry that we've been talking about. Antivirals, all right? That there's a whole class of drugs there. We've been able to target flu and AIDS with drugs. Yes.
So Can we go into a little bit of the instrumentation of how you can look at molecules in somebody? Like how can we as scientists look inside the brain, for instance, to see the neurotransmitters and how they act?
Right. So a lot of that is you can. So if you're actually watching the drug in somebody's body, I'm not actually too sure about how that works, but I do know, for example, how we actually assay the drugs in human bodies. So what we typically do is we can take these cells and we can actually build the machinery necessary. All right. So we basically have the cells like a Chinese hamster cell, I think is one. There's all kind of different cells that we use, and they express these neurons and so forth, and then we have them basically in a little vial all of a sudden, right? And then what we can do is we can start watching the drugs. We can take the drug and then the natural substrate, and we can put them in there and let them compete, and then we can wash away all the bio stuff and see what we have left. If we have a lot of the natural substrate left, that means the inhibitor did a good job, right? If we have a lot of the inhibitor left, that means it didn't do such a good job in all the all the natural substrates in the biological matter.
And then we had a question that says, if GABA shortage causes epilepsy, what would an overabundance of GABA cause?
Yeah, so GABA has this nice balance between glutamate and GABA, right? So an overabundance of it is just going in the other direction. So GABA essentially is what we call the inhibitory neurotransmitter. It's basically your off switch, right? So if you don't have enough of it, you're getting what we call overstimulation, right? And if you have too much of it, you're getting understimulation. So you want to have just the right amount.
And so it sounds like a lot of the organic chemistry, or at least the synthetic chemistry that goes into what you do, or making drugs for these diseases, is knowing how your compounds act with each other and then using that to your advantage to create new structures, right?
Correct.
Okay, very cool. Was that the end for you?
Yes, yeah, sorry. That was my last slide. And again, I just wanted to talk about, there was a lot of cool, hopefully cool biology looking at, kind of anti-Alzheimer's, just even looking at antivirals. And just, we don't want to get lost in what, an organic chemist actually does. We're the ones that synthesize or make these compounds, right? And I had I was very fortunate when I was in graduate school to work alongside a pharmaceutical company during my development of some of these compounds. And what you get to see is this, I guess, interdisciplinary work that goes on, right? So you have your team of organic chemists; those are your synthetic folks, right? Then you have some analytical chemists, and what their job to do is help you purify the compounds, make sure they're the correct compounds, and so forth. And then you have a very large team of toxicologists, biologists, biochemists, and they're all looking at different aspects of what you're doing, right? Is this compound doing what it's supposed to be doing, first of all, but is it doing things we don't want it to do, right? And then, of course, there's the marketing side of it. How much is this going to cost to make, and, things like that. So basically, an organic chemist, we're part of the team that is necessary to develop drugs. But we're kind of the first line where we just say, all right, can you make this compound, right? And once that compound is made, then it goes up the ladder and then it gets tested. But it's a long journey from a synthetic laboratory like I work in to a drug that's in a bottle.
Right. It always reminds me of when people say that biology is really just chemistry and chemistry is really just physics.
Yeah, they're all interrelated.
Yeah, absolutely. And do you think that your first grader and second grader are going to be synthetic chemists?
Well, we'll see. My first grader, now she's going into second grade. She's quite curious into chemistry, and she likes to make potions at home, which can kind of irritate mom and dad a little bit. Sometimes they involve food coloring and hand sanitizer, which is a little hard to come by now. But so she's definitely into mixing things, and we'll see. But she definitely has a scientific mind.
Great. And we have one more question that says, I've always wondered, how accurate are those diagrams to represent what's actually going on in our cells? Are they close to a magnification of the real things, or do they just represent what's going on chemically?
Okay, so yeah, we have two different things where I'll go ahead and share my screen again. And let me, it's a great question. All right, so when you look at something like this is not a cartoon. So this is actually using very complex microscopes and so forth. And what they can do is they can put false coloring on them, of course. But you know, what we're seeing there is, you know, real stuff. All right, so these are not cartoons. But if we look at something like that again is what we call an X-ray crystal structure. All right, so what we're doing there is we make a crystal of the ligand with the enzyme. All right, and then we can visualize it using X-ray crystallography. All right, so this, while it is a computer rendering, this is actual real data. So this is what the compound does look like. Now, this here again would be from an X-ray crystal structure. All right, so this is real, whereas this is what we would call a cartoon. All right, so what we're doing here is we can actually know what it looks like from maybe X-ray crystal structures and put that together. And things like these neurons and so forth, that's probably done with just simple microscopes. But you know, we kind of draw them in cartoon fashion.
And then before we end, I always like to ask my chemists, what is the biggest lab fail you have ever had?
Oh, biggest lab fail. There's so many. Let's see. I'm trying to think. I haven't had any explosions, so that's a good thing. And had very few accidents, so it's usually not that. But I'd probably say it's kind of nerdy. But one time I was working with a palladium compound, and palladium compounds are very expensive. And this compound happened to have an extremely high molecular weight, and I didn't need much of it to synthesize. I didn't need to synthesize much of it, but I wasn't being careful with my math, and I ended up making quite a bit of it. All right, that's probably still sitting in that lab down in North Carolina, and it probably cost me a couple $100. So I don't know if my advisor from NC State's listening or not, but I wasted a lot of palladium that day. So yeah, that was a bad one.
Yeah, we've all been there in the lab, so I'll leave everyone off with that little tip of just being extra careful in the lab.
Yeah, right. Double check your math.
Yeah, definitely. Thank you so much, Dr. Monceaux, for spending your morning with us.
No problem, and thanks for setting this up, Abby. It was fun.
All right, talk to you soon. Bye-bye.
Bye-bye.